Good afternoon to those of you who are expecting Judith Agudo today. I'm sorry to disappoint you. Judith is not feeling well and asked me to please step in to introduce our speaker for the afternoon. It's my honor and privilege to introduce Associate Professor Omer Yilmaz from the Whitehead Institute at MIT, Department of Biology at MIT, and the Department of Pathology at Mass General. Omer received his undergraduate and graduate degrees, both MD and PhD, from the University of Michigan in Ann Arbor. So we are fellow alumni of a distinguished institution. Omer did his PhD research in the lab of Sean Morrison and was instrumental in some of the very early findings in Sean's lab showing hematopoietic niche components in the endothelium as opposed to osteoblasts. The SLAM receptors and other beautiful biology that flowed out from that work began in part under Omer's watch. After that, Omer came here to Boston, Cambridge, and was a postdoc in David Sabatini's lab, where he did really elegant work on the metabolism and dietary control of the Paneth cells, which are a cell type in the stem cell compartment of the intestine and are thought to represent an integral part of the niche that supports that stem cell self-renewal. Omer then went on to join the faculty at the Cancer Institute at the MIT, the Koch Center, and has continued to do really very, very elegant work on stem cell biology and its relation to metabolism, to aging, to cancer. He is well known to many of us at Dana-Farber for his collaborative and interactive roles here. And it's a pleasure to introduce his talk titled Dietary Control of Intestinal Stem Cells in Physiology and Disease. Omer?
Ramesh, thank you very much for the kind introduction, and I'd like to thank Judith for the kind invitation to present, and I hope she feels better. And it's always a pleasure to present to colleagues at the Dana-Farber. So as Ramesh said, today I'm going to talk about work that we've done investigating the effects of diet on intestinal stem cells and how this leads to diseases such as cancer. But I decided to just give a brief overview of some of the work that we've recently done and really focus on two unpublished stories. So I apologize in advance if my presentation is a bit choppy, but this is really the first time that I'm going to speak about these two unpublished stories, and I look forward to recommendations and your thoughts. So I'd just like to start off and thank members of my laboratory, both past and present. It's really their hard work and creativity that I'm going to talk about today. And in particular, I'm going to focus on work by Nori Goto, a postdoctoral fellow in the lab who's really played an instrumental role in my lab in our understanding of how the stromal niche for intestinal stem cells responds to diet and aging, as well as understanding mechanisms of metastasis for colorectal cancer. So when I initially started my lab back in 2014, we really focused on understanding the link between obesity and cancer. And we've known for many decades that obesity is the modifiable risk factor for many cancers in the body, including for the colorectum, which is the tissue that we study in my lab. And not only is obesity a risk factor for cancer incidence, but it's also associated with poorer outcomes and cancer mortality for these different cancers of the body. So we became really interested in asking a simple question: how does obesity impact colon cancer incidence and progression? So colon cancer is the second leading cancer cause of death for men and women. So in 2020, in the United States, about 150,000 new cases were diagnosed. And unfortunately, about 50,000 patients succumb to complications of colorectal cancer. And many of these cases arise from metastatic disease, which I'm going to talk about in the second half my talk today. And an interesting observation that's becoming more apparent over the past decade or so is that the incidence of colon cancer in young patients, namely those patients less than 50 years of age, has increased dramatically in the past decade, raising an important role for diet and obesity in this process. So notably, the shifting of the incidence curve for colorectal cancer to younger and younger ages coincides with the epidemic of obesity in adolescents and young adults. And as a GI pathologist, I remember, as a fellow and as a resident, it was very infrequent that one would diagnose colorectal cancer in a patient younger than 40 years of age. And I have to say now, when I do service weeks, it's pretty common to have one or two patients with advanced colorectal cancer that are in their 30s and 40s. So our model system for addressing these types of questions is the mammalian intestine. So the intestine is a simple epithelium, meaning that it's a single layer of epithelial cells that serve as a barrier from the intestinal lumen to the body. And in the intestine, one has these crypt domains, and at the bottom of these crypt domains are the stem and progenitor cells. So the stem cells here denoted in green maintain the intestinal epithelial lining. They give rise to progenitor cells, which have lost the ability to self-renew, but maintain the ability to differentiate into the various differentiated and specialized cell types of the intestinal lining. And the intestine, like many adult tissues, is maintained by a hierarchy of primitiveness. The apex of primitiveness are the intestinal stem cells. These intestinal stem cells give rise to these progenitors, which have lost the ability to self-renew. And these progenitors undergo massive expansion and differentiation into the various intestinal epithelial cell types that make up the intestinal lining. So why focus on intestinal stem cells and this connection to diet and cancer? So it turns out that the intestine is a rapidly renewing organ. The average 70 kilogram adult generates about 100 to 150 grams of new intestinal tissue every day. Although most of the intestinal epithelial cell types have a very short lifespan on the order of 3 to 5 days, these stem cells shown here in green live for the life of the organism. So they persist for a very long time, and eventually a subset of them may accumulate oncogenic mutations and engender stem cell initiated cancer. So in the lab, we've been really interested in really dissecting out how stem cells respond to diet and how some diets predispose stem cells to initiate these tumors, whereas other diets such as caloric restriction and fasting dampen this tumor program. And model systems used in my lab and by many others, including at the Farber, include the use of LGR5 GFP Cree-EIT2 mice. So pioneering work by Hans Clevers has demonstrated that the vast majority, if not all, intestinal stem cells in the mammalian intestine express a gene called LGR5. And using these reporter mice, one can label these LGR5 positive stem cells with GFP. These mice are very important because, number one, they permit one to quantify the number of intestinal stem cells in response to different physiologies and diet. And number two, the presence of this Cree recombinase that's inducible with tamoxifen enables one to test what happens when genes or pathways are inhibited or activated in response to these diets. So a common assay that we use in the lab are to test stem cell function, our organoid assays. So one can isolate these GFP LGR5 positive stem cells, place them into three-dimensional matrix, and in the presence of growth factors such as Wnt, Notch, arspondin, and BMP inhibitors, these stem cells can engender these intestinal organoids or mini intestines in culture, which have a histology reminiscent of the in vivo biology, where there are these cryptodomains with stem cell at their base and zones of differentiation that are reminiscent of the upper crypt and villus domain of the small intestine and colon. So we use, for example, organoid forming capacity as a surrogate of intestinal stem cell function. In my lab, we've also been able to leverage organoid technologies to test the effects of diet and specific nutrients and metabolites on intestinal stem cell fate, work that in part we've published on, but that we're actively pursuing in the lab, testing out different amino acids and lipids. In addition to these types of organoid assays, my lab has also developed tools to study colon cancer. So work from geneticists such as Bert Vogelstein and others have shown that in order to convert normal intestinal mucosa into an invasive cancer that can metastasize to distant organs such as the liver, requires a sequential loss of tumor suppressor genes such as APC, where APC is mutated in up to 85% of all colorectal cancers, as well as subsequent mutations in oncogenes such as KRAS and loss of P53, activation of PI3 kinase, and many, many other mutations. And in the lab, we've been very interested in understanding how obesity and other diets regulate the evolution of tumor at these various stages of development. So in the early part of my lab for the past 6 or 7 years, we've been most focused on understanding how these diets such as obesity prime intestinal stem cells to go down this cascade of tumorigenesis, leading to invasive metastatic cancers. So how does obesity impact intestinal stem cell biology? Before I get into that, I'd just like to say that when Jayton Roper, a previous postdoctoral fellow in the lab who now runs his own laboratory at Duke Medical School, he developed a toolkit that permitted us to physiologically model colon cancer in mice. So using mouse colonoscopy, Jayton was able to release payloads of cells or colon cancer organoids into the lamina propria and submucosa of the recipient colon. After a period of time, these injections yielded not only primary tumors, but in a few weeks, a subset of these mice developed liver metastases. And really the study of liver metastases in the colorectal cancer field has been a challenge. And we're very excited to leverage this technology to gain more insights into the mechanisms and metabolism of metastasis. And I'm going to talk about this in the second half of my presentation today. So as I mentioned, this is a very important toolkit as it enables the rapid modeling of tumor initiation, progression, and metastatic disease. So what is the role of diet and obesity in the early steps of intestinal tumor formation? To address this question, my lab has looked at different types of obesity diets, and really a workhorse diet that we exploit in the lab is a high-fat diet. And in this high-fat diet, the mice derive about 60% of their energy from animal fat, 20% from protein, and 20% from carbohydrates. And when one ages these mice for about 18 months on a high-fat diet, not only do these mice become obese, but a subset of these mice develop spontaneous intestinal tumors. So here we're looking at the intestine from the outside. This bulge represents an invasive cancer. Histologically, these cancers are often moderately differentiated adenocarcinoma and reflect what one finds in patients. And when one quantifies, one finds that the high-fat diet leads to about a 3 to 4-fold increase in the incidence of spontaneous colorectal cancer. So in mice, we see not only does one find colon cancer, but one also finds intestinal tumorigenesis in the small intestine. And this work was really a collaboration between Samir Baz, a graduate student, Mieko Mana, a postdoc, and Jayton Roper, where Samir and Mieko now also run their own labs at Cold Spring Harbor and Arizona. So what happens to stem cells in a high-fat diet, obesity diet? So just to summarize a lot of work that we've done in this area, what one finds that in the high-fat diet obesity model, there's greater numbers of intestinal stem cells at the bottom of the crypt. So there's an expansion in the LGR5 positive intestinal stem cells. In addition to having stem cells with greater self-renewal and stemness properties, one also finds that progenitor cells that are higher up in the intestinal crypt acquire functional attributes of stem cells. So remember, stemness is a functional definition. And what I mean by this is that progenitor cells from the high-fat diet, like stem cells, are able to initiate intestinal organoids in culture, whereas progenitors from control diets are unable to do so. So we think that that molecular rewiring that occurs in the high-fat diet does confer stem cell-like attributes to these progenitor cells. And what we believe is happening here with respect to cancer is the following. So in normal dietary conditions, as I mentioned to you, stem cells are long-lived, and it's believed that they give rise to most colon cancers under normal physiologic conditions, where they accumulate oncogenic mutations, which allows them to initiate cancers. In the high-fat diet, the picture becomes a bit more complex. So in the high-fat diet, one finds that, number one, there's greater numbers of stem cells, and number two, progenitor cells can also behave like stem cells. And what we've shown occurs is that in the high-fat diet, one can have both stem cell-initiated as well as progenitor-initiated intestinal tumors. And mechanistically, what we propose is happening here is that, again, in the normal intestine, stem cells can serve as a cell of origin to give rise to tumors. And in the high-fat diet, what we propose is happening here is that lipids in the high-fat diet can stimulate these nuclear receptors called PPARs. And these PPARs can activate a very distinctive metabolic program in stem cells and progenitor cells that leads to the activation of fatty acid oxidation, and downstream of fatty acid oxidation, ketogenesis. And it's really this molecular program that leads to both stem cell and progenitor cell initiated tumor formation. And from a therapeutic standpoint, what one finds is that these tumors that arise in the high-fat diet are exquisitely sensitive to fatty acid oxidation inhibition. So this led us to ask the question, are PPARs and this fatty acid oxidation program the sole mechanism by which a high-fat diet leads to tumor formation, or could there be non-overlapping mechanisms that also contribute to intestinal tumor genesis? That is, are there non-PPAR mechanisms that contribute to the effects of a high-fat diet on intestinal tumor formation? So I'd just like to summarize and say that, this is all published work, some of it quite recently from late last year. And for those of you interested, please look at our papers. But I just want to summarize and say that we think that one mechanism by which a high-fat diet leads to greater tumor formation in the intestine is by engaging this PPAR fatty acid oxidation program. And a separate mechanism by which these stem cells are able to give rise to tumors independent of the PPARs is by downregulating immunomodulatory genes such as MHC class II. And the importance of downregulating immunomodulatory genes such as MHC class II enables premalignant stem and progenitor cells to evade immune cell detection. So we think that a high-fat diet model of obesity contributes to colon cancer through distinct pathways. One pathway is that the high-fat diet obesity, by engaging this metabolic PPAR fatty acid oxidation program, drives tumor formation. And in parallel, we've shown that a high-fat diet, by changing the microbiome, enables stem and progenitor cells to evade immune cell detection. And this also contributes to elevated tumor formation in a high-fat diet. One thing that we've become interested in over the past couple of years is what effect does the high-fat diet obesity have on the stromal intestinal stem cell niche? So we know that intestinal stem cells located at the bottom of the intestinal crypt receive various trophic factors from neighboring cells. So here we're looking at an intestinal stem cell at the bottom of the crypt that is adjacent to a panicell. So these adjacent epithelial panicells can elaborate growth factors such as Wnt and and Notch ligands that activate two important pathways that are needed for stemness, that is Notch and Wnt. In addition to these epithelial-derived cues, it's been now well understood that there's also stromal-derived cues, such as our respondins and BMP inhibitors, that play an important role in maintaining the stem cell program in intestinal stem cells at the bottom of the crypt. And work from Ramesh's group and others have really beautifully delineated the many different stromal cell populations in the intestinal mesenchyme that contribute to intestinal stemness. So there's many different cell types, such as telocytes, trophocytes, myocytes, pericytes, endothelium. All of these cell types are to some degree or another in close proximity to the LGR5 positive stem cell at the bottom of the crypt, yet it's unknown, for example, which of these cell types is the dominant source of our spondin, which really is an LGR5 specific factor that drives the stem cell program. And what I mean by this is that there's actually in the literature there's some uncertainty regarding the stromal source for our spondins for intestinal stem cells. So I'm only going to focus on our spondin 3. So our spondins are a family of 4 members, but our spondin 3 is really the most highly expressed our spondin family member in the small intestine and colon. And there's been a smattering of publications, some suggesting that telocytes are the main source of our spondins, others showing that lymphatic endothelial cells produce our spondin, but this paper provides no functional evidence. And really nice work from Neil McCarthy in the Shivdasani lab, demonstrating that a subset of fibroblasts called trophocytes are an important source of our spondin. So we really wanted to take a step backward and really focus on identifying what is the main source of our spondin. And the reason for this is we reasoned that if we could find the main cellular populations that provide our spondin to LGR5 positive stem cells, those cellular populations may play an important role in coupling the ability of diet to communicate to the LGR5 positive stem cells, given that our spondin is the ligand for LGR5 itself, which is really a defining marker for intestinal stem cells. So Nori Goto, as I mentioned in my introduction, is a fellow in the lab, and he took a genetically engineered mouse approach. So he obtained our spondin 3 GFP back transgenic mice, and in data that I'm not showing you, we've also generated our spondin 3 TD tomato pre-ERT2 knock-in mice, and I'm only going to focus on the back transgenic mice because for the most part, we see complete agreement between both of these genetic approaches. And what Nori observed is that our spondin 3 seems to be expressed by these slit-like spaces near the bottom of the intestinal crypt, both in the small intestine and in the colon. And to a pathologist, these spaces look like vascular channels, such as lymphatic channels. And indeed, that's what one finds here. So if one does co-localization with LIVE1, which is a lymphatic endothelial specific marker in the R-spondin 3 back transgenic mice, one finds nice co-localization between LIVE1 and GFP at the bottom of the intestinal crypt, again, in the small intestine and colon. We went ahead and wanted to understand, well, are lymphatic endothelial cells in close proximity to many intestinal crypts, or is it really a patchy phenomenon that one captures in a particular field of view? So here we generated a modified version of the LGR5 GFP reporter initially generated by Hans Cleavers. This new allele has a GFP CRE in the 3' UTR, where the GFP is now expressed in all the LGR5 positive cells of the intestinal crypts. And using these new knock-in mice, when one stains for LIVE1, one finds that LIVE1 positive lymphatic endothelial cells are indeed in close proximity to many of the intestinal crypts. And when one quantifies this, one finds that greater than 80% of intestinal crypts in the colon and small intestine are essentially juxtaposed to these lymphatic channels. And this isn't some idiosyncrasy of mice, because in human sections, such as in human duodenum or in human colon, one comes to similar conclusions. So here's confocal microscopy for mice. So green are the LGR5 positive stem cells, and red are the LIVE1 positive lymphatic endothelial cells. And again, one finds that these LGR5 positive stem cells are essentially juxtaposed to these lymphatic endothelial cells. So to gain better insight into what cell types express our spondin 3 within the intestinal stroma, we sorted out the R-spondin 3 GFP positive cells and performed RNA sequencing. And one finds that these R-spondin 3 positive cells really come in two flavors. So there's a subset of fibroblasts that express R-spondin-3, and lymphatic endothelial cells express R-spondin-3. And there's a very small subset of these stromal cells called telocytes, which were previously implicated to be a dominant source of R-spondin in the intestine. But in our analysis, at least this turns out to represent really a minor population. So previous work by Ramesh's group, so our data confirm work by Ramesh's group, where again, these R-spondin-3 gremlin-1 positive fibroblasts seem to be an important source of R-spondin-3. In addition, lymphatic endothelial cells are R-spondin-3 expressing. So we took advantage of our back transgenic mice and flow cytometry and sorted out lymphatic endothelial cells, and we call these R-spondin-3 gremlin-1 positive fibroblasts RG fibroblasts, and these essentially contain the trophocytes that were defined by the Shiv Dasani lab. And when we sort these populations out from the small intestine and colon and perform bulk RNA sequencing, we indeed confirm our single cell analysis where the R-spondin-3 positive CD31 negative fibroblasts are gremlin-1 positive and R-spondin-3 positive, whereas the CD31 positive, R-spondin-3 positive lymphatic endothelial cells are gremlin-1 negative and R-spondin-3 positive. And I should just mention that gremlin-1 is an important factor for stemness, as gremlin-1 is a BMP inhibitor, and I'm going to talk more about that in my next slide. So we wanted to get better resolution with regards to where these RG fibroblasts and lymphatic epithelial cells were with respect to the LGR5 positive intestinal stem cells. So we generated a gremlin-1 TD tomato pre-ERT2 knock-in allele and mated it to our R-spondin-3 GFP back transgenic. And what one finds here is that when one zooms into the bottom of the intestinal crypt, R-spondin-3 positive GFP marks lymphatic endothelial cells shown here with nice overlap with live one. So the overlap here is shown in white. But in addition to overlapping with live one positive cells as predicted from our single cell analysis, there's R-spondin-3 positive cells surrounding these lymphatic vascular channels that are gremlin-1 TD tomato positive. And in the overlap, these RG fibroblasts are shown in yellow. And this really leads us to the following model, both in the small intestine, which I'm showing here, but also in the colon, which is that the intestinal crypt base is in close proximity to lymphatic endothelial cells, and surrounding these lymphatic endothelial cells are these RG fibroblasts. And both of these lymphatic endothelial cells and RG fibroblasts are R-spondin-3 positive. So again, in data that I'm not showing, we have a similar finding in the colon, but what about function? So to address function, we performed both in vitro heterotypic co-cultures as well as in vivo deletion experiments. So we adapted protocols from Neil McCarthy to really ascertain how these two different populations supported the ability of intestinal crypts to form mini intestines in culture. So Nori sorted out the lymphatic endothelial cells and the RG fibroblasts using our reporter mice. He propagated the fibroblasts and lymphatic endothelial cells in culture. Again, these are primary cellular populations from the intestinal stroma. And then he added intestinal crypts to these fibroblasts or lymphatic endothelial cells and assayed the ability of these cellular populations to support the ability of these crypts to form organoids. So what Nori found is that in culture conditions that lack R-spondin, but include noggin, and noggin as a BMP inhibitor, what he found is that lymphatic endothelial cells are capable of supporting supporting crypts to form organoids. But RG fibroblasts are much more capable of doing so. And I just like to point out that we think that some of this difference between lymphatic endothelial cells and RG fibroblasts is somewhat of an artifact in that it's somewhat of a misrepresentation in the sense that RG fibroblasts really grow well in 2D cultures, whereas primary lymphatic endothelial cells do not. So we think that some of this difference is due to our inability to really robustly grow lymphatic endothelial cells. We also perform the same heterotypic co-culture in media conditions that lack not only our spondin, but also lack the BMP inhibitor noggin. And in these co-cultures, the only stromal population that was capable of supporting the ability of crypts to form these mini intestines are the RG fibroblasts, which makes sense given that these RG fibroblasts not only make our spondin, but they also generate gremlin 1, which can serve as a surrogate for BMP inhibitor noggin in culture. And this is quantified here at day 4, where RG fibroblasts are quite capable, and LECs are unable to support without a BMP inhibitor. So most importantly, what about in vivo? What is the in vivo source for our spondin 3, and which population, RG fibroblasts or lymphatic endothelial cells, play a dominant role? So to address this, we again took advantage of our genetically engineered mice. So to delete our spondin 3 in the RG fibroblasts, we use our gremlin 1 CRE-ERT2 mice. And to delete our spondin 3 in lymphatic endothelial cells, we obtained PROX1 CRE-ERT2, or PROX1 is a specific CRE for lymphatic endothelial cells in the intestine. So we generated our spondin 3 gremlin 1 mice, our spondin 3 conditional PROX1 CRE mice, and then we generated the compound mice where one is able to delete our spondin 3 both in the lymphatic endothelial cells and in the RG fibroblasts. So we administered tamoxifen and analyzed 3 days after the last dose of tamoxifen administration. So what does one find? So what we found is that when we delete our spondin 3 in either the lymphatic endothelial cells or in the RG fibroblasts, the overall morphology of the intestine is normal. So there's normal crypt-villus length. There's no increase in apoptosis. One important finding that we did find perturbed when one deletes our spondin in either of these populations is that there is a reduction in the number of stem cells, quantified here with 2 markers for intestinal stem cells, that is olfactomedin 4 and LGR5. Both of their expression goes down upon loss of our spondin 3 in either of these populations. Interestingly, when one deletes our spondin 3 in both the lymphatic endothelial cells and in the RG fibroblasts, this now leads to shorter villi crypts and to a more dramatic loss in olfactomedin 4 and LGR5 positive stem cell numbers, which is quantified here to the right. And again, similar effects are seen in the colon. So what about in injuries? So in homeostasis, loss of our spondin 3 in both the RG fibroblasts and lymphatic endothelial cells seems to play an important role. So what about with injury? So to injure the intestine, we administered 12 grays of irradiation. And again, the control intestine is quite capable of repairing itself. When one deletes our spondin in either the RG fibroblasts or in the lymphatic endothelial cells, now the small intestine is significantly impaired, and in the crypt, colonic units become a little bit shorter. But when we now lose our spondin 3 in both of these cellular populations, the small intestine is essentially obliterated. It becomes essentially completely ulcerated, as does the colon. And the colon phenotype is quite interesting, given that the colon is much more radio-resistant than the small intestine. So we think that our spondin 3 loss in both lymphatic endothelial cells and RG fibroblasts compromises intestinal integrity and plays an important role in stem cell mediated repair. And I'd just like to point out there are benefits to being a pathologist, because when one looks at these slides, something really catches your eyes, which is after radiation, there's a clear expansion in lymphatic channels. So these dilated vessels at the bottom of the intestinal crypt are actually lymphatic channels that become dilated after intestinal injury with radiation. So to quantify this, Nori performed flow cytometry, where he quantified the R spondin 3 positive lymphatic endothelial cells and the RG fibroblasts. And what one finds is that after radiation, there's about a 2-fold increase in both of these populations. So there's clearly an expansion in these 2 important stromal niche components for the intestinal stem cells. We also performed RNA sequencing, single cell RNA sequencing on these respondin 3 positive stromal cells after radiation. And again, akin to what one finds in the control state with radiation, respondin 3 positive cells are mostly the RG fibroblasts and the lymphatic endothelial cells. And again, we have this small telocyte population. And interestingly, not only is there an expansion in the numbers of lymphatic endothelial cells and RG fibroblasts, but qualitatively, these lymphatic endothelial cells and RG fibroblasts generate greater levels of respondin. So they're boosting production of respondin, which is an important trophic factor that drives the intestinal stem cell state. And mechanistically, what we think is happening here is that after radiation, the RG fibroblasts, for example, strongly activate the IL-1 receptor, which is barely expressed under control conditions. And this is consistent with GSEA analysis, where the RG fibroblasts show a very distinctive IL-1 mediated signaling pathway. And to understand what role IL-1 might be playing in the irradiated RG fibroblasts, Norrie isolated the RG fibroblasts, placed them in culture, and exposed them to IL-1A. And what he found is that exposure to IL-1 led to about a 2-fold increase in respondin 3 production by these RG fibroblasts. So I just like to summarize this part of my talk and say that under normal homeostatic conditions, we believe that RG fibroblasts and lymphatic endothelial cells support intestinal stem cells through the production of respondin and gremlin-1. And in data that I didn't have time to talk about, it actually turns out that RG fibroblasts and lymphatic endothelial cells can regulate one another. So RG fibroblasts secrete trophic factors such as VEGF-C and D, which are very important for maintaining LEC. So if one ablates these RG fibroblasts with diphtheria toxin, this leads to a massive reduction in the lymphatic endothelial cells. Similarly, we've identified a factor secreted by LECs called relin. And relin, when relin in culture, relin drives the expression of respondin 3 in these RG fibroblasts. In regeneration after radiation-induced injury, these pathways go into hyperdrive, where there's now expansion in fibroblasts and lymphatic endothelial cells, which really boosts respondin 3 production, permitting repair of the intestinal lining. So we're really excited by these findings because we think that there's this interesting dyad in the intestinal stroma between these lymphatic endothelial cells and RG fibroblasts that play an important role in regulating LGR5 stemness. And now we're really interested in using these genetic models that we've developed to identify these populations to really ask different questions, such as how do these stromal niche cells respond to diet-induced obesity and other physiologies such as aging. And we believe, for example, in aging, age-related changes in these RG fibroblasts seem to have a very prominent role in regulating LGR5 aging phenotypes. So with that, I'd like to kind of transition to some of the work that we've done with respect to colorectal cancer metastasis. If you remember, I mentioned that colon cancer is driven by a sequential acquisition of somatic mutations where one is able to convert normal mucosa into an invasive cancer that can metastasize. And early on in my talk, I shared with you work that we've done that really outlines how obesity alters the biology of intestinal stem and progenitor cells to induce tumor formation. And now we've become very interested in really the other end of the spectrum, which is invasive cancers and metastasis. How does obesity alter tumor progression and metastasis? And we've known from a lot of sequencing studies that there's really no gene mutation that distinguishes metastasis from primary tumor in colon cancer, really highlighting an important role that epigenetic changes play in this process of metastasis from the primary tumor to distant organs. So my lab has spent a lot of time developing organoid models for colon cancer. So Jonathan Braverman, a postdoc in the lab, has generated an isogenic series of mouse and human organoids that lack, for example, APC, KRAS, P53. So this combination that I call AKP is a very commonly seen combination of mutations in colon cancer. So using CRISPR-Cas9 and genetically engineered mouse tissue, we can generate naive AKP organoids in culture. And what I mean here by naive is that these AKP organoids have never been a cancer in vivo. They've only been cancers in the in vitro setting. One can then transplant these naive AKP organoids into the colon of recipient mice and assay their ability to spawn primary tumors and seed liver metastases. And using these AKP naive organoids, what one finds is that, and I should also mention that these organoids are labeled with TD tomato are labeled with TD tomato. So one can appreciate that these recipient mice, develop very large primary tumors after a few weeks, and a subset of these mice go on to develop liver metastases, where one can appreciate the formation of these TD tomato foci in the liver. So on average, these naive AKP recipients live about 80 days. And for the rest of my talk, I'm really going to focus on this AKP phenotype. But an important question that I'm not going to talk about today is, what about other genotypes? And we are very interested in understanding how different genotypes mediate some of these observations. But these AKP recipients on average live about 80 days, and about 30% of them develop liver metastases. And the mice that develop liver metastases develop a range anywhere from 1 up to 10 lesions. And this percentage of 30 is consistent with what we've previously published. So, we think that at least in our hands, that naive AKP on average will met about 30% of the time. So we became really interested in the following experimental paradigm, that is, can one generate pro-metastatic mouse and human AKP colorectal cancer organoids? And the idea here is that one takes naive AKP organoids, transplant them into recipient mice. A subset of these mice will develop liver mets, and then can one do forced evolution? So can one take these liver mets, generate organoids, and retransplant them back into the colon, take primary tumor organoids, transplant them back into the colon, do this iteratively until one generates pro-metastatic organoids? So is this possible? Indeed it is. So when we look at the percentage of liver mets derived from our primary and secondary organoids, or from our primary and secondary mets, as we passage the mets in vivo, they become more and more metastatic to the liver, whereas the primary tumors really don't show much metastatic potential. And the number of mets actually increases with passaging. So compared to the primary mets, there seems to be a trend towards greater number of mets per recipient mice. So we became interested in understanding, how does the chromatin accessibility and transcriptome change in these pro-metastatic organoids? Can we identify a pro-metastatic gene signature in this isogenic transplantation model? And I should mention that, at least in culture, primary and metastatic AKP organoids behave similarly. So in order to find differences, one needs to really do these in vivo transplantations. So by RNA-seq and ATAC-seq, interestingly, the different passages tend to cluster together. So by ATAC-seq and by RNA-seq, the naive AKP organoids cluster together. And by the third passage, primary, tertiaries, and primary mets by ATAC-seq and RNA-seq seem to form distinct clusters by PC analysis. And with the help of a bioinformatician in Tyler Jack's lab, we were able to identify in vivo signatures and pro-metastatic signatures from the transplanted recipients. And just to point out, if one takes our pro-metastatic signature and looks at its expression in the TCGA analysis for colorectal cancer, one finds that tumors that have a high pro-metastatic signature seem to have lower overall survival. So we wanted to spend some time integrating our RNA-seq and ATAC-seq data, and this is one ends up with a lot of interesting targets to pursue. But Nori was very interested in going after transcription factors, and he became very interested in SOC17 because not only does SOC17 show up in the in vivo signature, it also shows up in the pro-metastatic signature. And we were interested in SOC17 mainly because there's really not much known about SOC17 and its role in colorectal cancer progression and metastasis. So there's some work, for example, showing that SOC17 can mitigate Wnt beta-catenin signaling, but the mechanism is unclear. And a paper last year in Nature Cell Biology proposed that SOC17 in lung cancer cooperates with LKB1 mutations to drive metastasis. But what's happening in colorectal cancer? So to start to get the mechanism, Nori generated SOC17 knockout AKP organoids and engrafted them into black 6 mice. So he got successful deletion with CRISPR and then transplanted control and SOC17 knockout AKP organoids into the colon of mice and analyzed the mice 6 weeks later. And surprisingly, what Nori found is that a loss of SOC17 really inhibited the ability of these AKP organoids to engraft into the recipient colon. So whereas the control organoids are quite capable of engrafting, 80% of them form large primary tumors and metastatic lesions, the SOC17 knockout organoids really barely engraft in these recipient mice. Only less than 10% or so are capable of engrafting. And of course, because of low engraftment, the SOC17 knockouts do not give rise to metastatic lesions. So we looked at the histology of the tumors, and consistent with what's been previously shown and what we know from human pathology, that KRAS mutant tumors are often immune cold. So when we stain for CD3, 8, and CD4, there's really a very low frequency of these T cell populations and very low cleaved caspase-3. However, when one looks at this from the SOC17 knockout AKP organoids, one now finds a big increase, for example, in cleaved caspase-3, and now there's a much stronger infiltrate of CD3, CD8, and CD4 positive T cells within the stroma of the tumor. So we were really excited when we found this. We were both excited and a little cautious because we're not immunologists. So we reached out to our friend at the Farber and collaborator, Judith Segudo, who's been really a great colleague, and it's been great to collaborate with her. And we performed, so what we think is happening here before I get into the next set of data is that SOC17 seems to be important for in vivo tumor progression and perhaps metastasis, and perhaps it is so by regulating tumor immune cell interactions and perhaps permitting T cell evasion. So with Judith's advice, we went ahead and transplanted SOC17 knockout AKP organoids into immunodeficient mice. We transplanted both into RAD2 mice, which I'm showing here, and into non-skid gamma mice, which I'm not showing, but comes to the same conclusion. And what one finds here is that now the SOC17 knockout organoids are quite capable of engrafting into these immune compromised recipients. So now 100% of both the SOC17 knockout and control organoids are able to engraft into the recipient colon to form primary tumors. Not only are they able to form primary tumors, but interestingly, the SOC17 knockouts, although they form primary tumors, don't seem to give rise to liver mets. So there seems to be an uncoupling. So SOC17 in immunodeficient mice is able to form a primary, but SOC17 within the tumor itself seems to play an important role in metastasis, which we're not seeing in these immune compromised recipients. And again, to really nail this down to T cells, we performed the following experiment. We performed CD8 T cell depletion experiments where we essentially conditioned recipient mice with injections of CD8 antibody to deplete this population of T cells, transplanted the knockout organoids and analyzed the tumors 4 weeks later, and find that when we deplete CD8 positive T cells, as we observed in the immunodeficient mice, almost 100% of these injections led to engrafted tumors in the knockout condition. So we think that these SOC17 knockout organoids play an important role in interacting with CD8 positive T cells, but what's the mechanism? What's happening in these CD17 knockout tumor cells that's controlling the ability or encouraging these CD8 positive T cells to home into the tumor microenvironment to mount an anti-immune response. So we went ahead and performed RNA sequencing on the SOC17 knockout and proficient AKP organoids. And one pathway that really jumped at us were genes involved in the interferon pathway. So interferon response genes are upregulated in the SOC17 knockout AKP organoids. So here we're looking at individual candidates as well as by GSEA analysis. And if one stains, for example, for phospho-STAT1, which is downstream of the interferon receptor, in the SOC17 proficient tumor cells, there's very little phospho-STAT1 staining, in contrast to the SOC17 knockout tumor cells, where one can appreciate nice positivity. In addition to interferon responsive elements, interestingly, one finds induction of genes involved in pyroptosis, such as gasdermin D, which is an effector of pyroptotic death. So pyroptosis is an inflammatory type death. So upon activation of the inflammasome, this leads to the activation of caspase-3, which can generate mature IL-1, as well as generate an active form of gasdermin D, which can form these pores, which allow the spewing of cytoplasmic components into the extracellular space. And this was interesting because when one stains these tumors with gasdermin D, one finds that the SOC17 knockout tumors show a very strong induction of gasdermin D protein. So clearly, both at the RNA level and the protein level, gasdermin D becomes strongly activated and induced in the SOC17 knockout organoids. And this was really exciting because work by Judy Lieberman's group in Boston, as well as by others, have proposed that pyroptosis induces an anti-tumor immunity, in part by releasing IL-1 into the extracellular matrix. This is a mechanism by which T cells and dendritic cells become activated and perhaps mount a better immune cell response. So to get to some mechanism, or to at least test this idea, Nori generated gasdermin D, SOC17 knockout organoids, and NLRP6. NLRP6 is a redundant component of the inflammasome. And what he finds is that when he knocks out gasdermin D, he's now able to partially rescue the ability of these SOC17 knockout organoids to engraft into the recipient colon. So I just like to summarize and say that we're really excited here. We think that SOC17 regulates tumor and immune cell interactions in colon cancer, where SOC17 turns on in the in vivo tumor, in contrast to the naive state in culture. When SOC17 turns off in vivo, this leads to an increase in the interferon response, as well as pyroptosis, which we think plays an important role in activating CD8 positive T cells, which can mount a better anti-tumor response. So the question that we're interested now is how. So we're now doing cut and run for SOC17, and we actually think that there's some interesting interaction between SOC17 and GATA6 with respect to this program. And secondly, concurrent with this work that Nori's doing, we've already got experiments, analogous experiments going on in different dietary conditions and in different genotypes to understand how obesity regulates this process, as well as how different genetic mutations commonly observed in colon cancer regulate the process of tumor progression and metastasis. So with that, I just again like to thank members of my laboratory. Today, I really highlighted the work of Nori Goto, but Swagata Goswami is doing similar work in the high-fat diet and with different genotypes. And Joseph Sedlak is also working on the high-fat diet angle here. I'd like to thank our great friends and collaborators at the Dana-Farber, Judith Segudo, who's really been fantastic to collaborate with on the SOC17 project, and with Ramesh's group for all of their help, suggestions, and expertise with regards to understanding what cell types are important with respect to our spondin production in the stromal niche of the intestinal stem cell. And with that, I'd be happy to take some questions.
Great. Thank you, Omer. That was a terrific talk. It really spanned the breadth of much of intestinal stem cell biology as it relates to cancer. So thank you again. I'll start with questions that are in the chat, in the Q&A box, and I encourage other attendees to use the Q&A box, not the chat feature in Zoom, to enter your questions, please, and I'll narrate those questions for the speaker. So Jonathan Frieden has two questions. One of them is very general, whether the effects of a high-fat diet are reversible. And the second, more specific question is that nuts are high in fat, but not animal fat. Does a diet in which most calories come from nuts promote the same effects?
Mm-hmm. Yeah. So the first question we're actively working on. So we're really tackling the first question using two approaches. The first is we're doing very simple reversal experiments. So what happens when one comes off of the high-fat diet? How much memory is retained, and how long does it persist? And does the latency of re-exposure to high-fat diet go down once you've entered that state previously? So we're actively pursuing that question by doing very simple reversal and re-exposure type diets. We're also actually looking at a model of gastric bypass. And what's interesting about gastric bypass is mice, when you perform gastric bypass in a high-fat diet state, they become lean, but they're still on the high-fat diet. And we're really interested to kind of uncouple, the diet from leanness and obesity itself. With respect to the second question, yeah, different fats are going to play different roles. And that's been a question that, we've been very interested in. I have to say, we haven't spent a lot of time, looking at, fats found in nuts, or, really looking at other lipids. Studying lipids is very challenging just because there's so many of them. But we are looking at other obesity diets, such as fructose-rich diets and glucose-rich diets, to kind of broaden our understanding of how different modes of obesity regulate these processes.
Thank you. So Raya Bekala has a question. Why do you think the PPAR signaling axis is not also regulating MHC class 2 downregulation?
Yeah. So we've done both. So when we treat mice with agonists for the PPARs, or when we overexpress the PPAR genetically, we see no perturbation in MHC class 2 levels, number one. And number two, in genetic models where we knock out all of the PPARs, we see no change in MHC class 2 expression. So, we think that's pretty convincing evidence that the changes we observe in MHC class 2 in response to changes in the microbiome are occurring independent of the PPARs.
And Kelly McGovern asks, do you see as much T cell infiltration in the Gasdermin D knockout tumors?
Yeah, so good question. I don't know the answer to that question. That data is literally like a week old, a couple of weeks old, but that's something we should look at.
Then there are two questions about SOC17. First, Raul Mostoslavsky congratulates you on a fantastic talk and asks whether expression of SOC17 is present in all metastatic cells or if it's heterogeneous.
Yeah, Raul, that's a great question. We've now got a SOC17 GFP reporter mouse. So we've now mated the SOC17 GFP reporter into our AKP alleles. So we're hoping to look at that very carefully. It's hard, by IHC, it's hard to look at SOC17, but we do think, probably there is some type of heterogeneity but I think this GFP approach is going to allow us to really precisely look at that question and to really dissect out whether SOC17 GFP high versus GFP low cells have different in vivo biology.
And then related to that, Sandro Spisak asks, how did you find SOC17?
Yeah, So yeah, SOX17, it's really interesting. Uh, I mean, SOX17 number one goes up dramatically. We got, in some ways, we got really lucky because um, these naive AKP organoids, as soon as you put them in vivo, SOX17 levels go up dramatically. And what we're really interested in, what is the signal in vivo that drives SOX17 expression? So just by doing simple RNA sequencing, we identified it. We had actually identified it a few years ago in other experiments where we were taking just naive organoids and transplanting them in vivo, but we never really pursued it until Nori took over the project and did this very careful iterative transplantation model. So just by gene expression, it's one of the most upregulated genes going from in vitro to in vivo.
And it's probably one of the few transcription factors, I presume. You said there was a particular
It is.
Transcription factor.
Exactly. And what's interesting is if you go from in vivo back to in vitro, it stays on. So once it's on, it's on. That's what's really cool because we did our RNA sequencing. We didn't do it from in vivo tumors. We generated organoids, propagated them in culture for 10 or 14 days, and then did our RNA-seq and ATAC-seq. So the signature that we're identifying is a maintained signature that seems to persist even in the in vitro setting upon removal from the in vivo environment.
All right, it's a little bit past the 5 o'clock hour. I know you and I have a little time to catch up later in the week. So
absolutely.
If there are no other questions from the audience, then we can close the forum. With many thanks again for taking the time to share your beautiful unpublished work with us. Thank you.
Well, thank you very much.
Thank you all for attending.